Good morning, dear student. Now we are going to discuss about few important questions related to chapter 1 biochemistry. Okay. So first come to first question. A polypeptide was subjected to the following treatment with the indicated results. Okay, acid hydrolysis. After acid hydrolysis we are getting this fragment. Acid hydrolysis give you idea about only composition. You cannot get any sequence idea. Then aminopeptidase M, which will cut at N terminal, okay, and you are getting no fragment. Carboxypeptidase A and B, which is going to cut at what? Your C terminal, and you are getting no fragment. From these two, you can get the idea it is a circular polypeptide, because these are exopeptidase. They will cut only at end. As you are getting no fragment after digestion with aminopeptidase and carboxypeptidase, it indicates that it is a circular peptide. Got the idea? From that you can get 4 is the answer. No need to go through 1, 2, 3. फिर भी you have to check whether your answer is correct or not with the help of fourth statement. So trypsin treatment give you your what? 2 fragment. Trypsin, that you have to remember, trypsin cut just after lysine or just after arginine at C terminal. So see here, so here one cut occur, here one cut occur. Okay, so you are what you are getting? So you are getting cysteine, glycine, leucine, phenylalanine, arginine, that is correct. And you are getting that is what? Threonine, alanine, methionine, glutamine, and your what? Lysine you are getting. That means which is the best answer? 4 is the best answer. Got the idea? Okay, so next. In mature RBC, erythrocyte, the end product of glycolysis that contain the carbon of glucose. So normally in case of what happen, end product of the glycolysis is the pyruvate. Okay, in the glycolysis, if it is it is having mitochondria, then it enter into mitochondria to produce what? Acetyl coenzyme A. But in this case, erythrocyte devoid of mitochondria. Mature erythrocyte is devoid of mitochondria along with a nucleus. So no questions of aerobic respiration. So that leads to lactic acid fermentation. So answer is lactic acid. Got the idea? Then during glycolysis in plant, alanine and related amino acids are directly coming from where? Now these are directly coming from which one? That is the pyruvate. Okay, pyruvate plus amino acid. Pyruvate plus amino acid, that will give you idea about, not amino acid, sorry, ammonia. Pyruvate plus ammonia give you idea what? That alanine. Okay, so next, in the context of protein import in nucleus, okay, which molecule is responsible for releasing the cargo from the importing receptor? So already we have discussed, okay, in our class, in case of nuclear protein transport or import is required, and inside that RAN GTP, okay, RAN GTP is helping for the dissociation of cargo. So here which protein is important? RAN protein. Okay, RAS, ROA, ROC, all these are signaling molecules, signaling relay proteins. Then which one of the following codons is used to code the selenocysteine? So selenocysteine and pyrrolysine, 21st and 22nd standard amino acids are coded by 2 terminating codon. UGA, that is selenocysteine, UAG, that is what? Pyrrolysine. Okay, so that you have to remember. Then out of following statement which is correct? Radioimmunoassay are more sensitive than enzyme linked immunosorbent assay or ELISA with chromogenic substrate, that is correct one. ELISA with chromogenic substrate are more sensitive than ELISA with chemiluminogenic substrate. No, chemiluminogenic substrate is more accurate and efficient than that of chromogenic substrate. So it cannot be. And ELISpot just like what? Same as that is your ELISA. Principle is exactly same. Only thing, okay. So here you are going to find out the thing or antigen present in one cell. You are not going to calculate the cell number. So it is given the cell number, so it cannot be. And Western blot, okay, so the analysis, the product of enzyme substrate reaction localized at the site precisely where the antibody enzyme conjugate binds to its specific protein band. Yes, if protein band is there, suppose your protein band is there, on that which is attached, on that your primary antibody will be attached, on that your secondary antibody will be attached. 1 minute. So your primary antibody, suppose this is the spot, so on that your primary antibody will be attached like this. On primary antibody, secondary antibody will be attached. Secondary antibody normally designed against FC region, and secondary antibody is along with what? The tagged with enzyme. So enzyme substrate, the complex will be for the complex of this substrate. The complex will be for on what? Antibody, antigen, antibody complex. So that is correct one. So D correct. Okay, 1 minute. D correct and A correct. Which is best answer? 2. A and D both are correct statement. So amino acid residue predominantly involved in protein DNA interactions. So protein DNA interaction means DNA is negatively charged, and so which protein should interact with DNA perfectly? Now protein is positively charged, so positively charged protein. Mostly it must be made up of mostly what? Arginine and lysine. Both are positively charged amino acid. Then another question, molar absorption spectra level I, 2I, 3I and given. So this is near about 280, this is near about 275, this is near about 260. Okay, actually the molar absorption coefficient or Amax of tryptophan is 280, tyrosine 275 and phenylalanine is 257. Okay, so which is the best answer? So 1A. Okay, so the data is given to you. Fluidity of phospholipid membrane phospholipid increases where the fatty acid. See here. So here concept of membrane fluidity. Okay, membrane fluidity increases. That means melting temperature of membrane should be less. So melting temperature of membrane or melting temperature of your, that is what? Fatty acid, that depends upon two things. One is chain length, which is directly proportional. One is unsaturation, which is inversely proportional. So if melting temperature I want to increase, then you have to increase the chain length, you have to decrease the unsaturation. But here it is saying that melting temperature increase. That means melting temperature increase means you have to increase the chain length and decrease the melting temperature. So here fluidity increase. Okay, here fluidity increase means melting temperature should be less. Melting temperature should be less. So melting temperature less only when the chain length decrease and the unsaturation should be increased. Then only melting temperature decrease, then only fluidity of the membrane will increase. So I think B is the best answer. Got the idea? Okay, in the next class we will discuss about rest part. Thank you.